DNA is a big, long, complex molecule that is found in all of your cells. This is just a small section of DNA. DNA extractions are used in labs all across the world every day as the first stage of onward investigations. I'm going to show you a very basic DNA extraction using everyday household reagents so that you can also try to do this at home as well. We're going to use a tool, thin glass, like a champagne flute, a beaker or a glass or a jar, really strong alcohol or methylated spirit, some salt, washing up liquid or liquid soap, a measuring cylinder or a measuring jug, a tea strainer, or you could use filter paper if you don't have a tea strainer, a chopping board and a knife. So I'm going to use a kiwi, but you could also use papaya or strawberries, whatever you can easily get hold of. So the first thing that we need to do is to take the skin off our kiwi or our papaya, and then once you've got the skin off, you need to cut it up as small as possible until it is almost like kiwi liquid, like kiwi juice. So this might take you a little bit of time and a lot of chopping. So once you've got your kiwi all finely chopped, you can take a glass or a jam jar or a beaker, whatever you have at home, and a measuring jug. I'm using a measuring cylinder, and you need about 100 millilitres of water. You put that into your beaker and then add 1 teaspoon of salt and give that a good stir, and 1 teaspoon of washing up liquid or liquid soap. And again, give that a stir, but try not to make too many bubbles with it. Then to that you can add all of your kiwi mash and kiwi juice, and again give that a stir. Now we want to keep this mixture warm, so I'm going to put this into a water bath that is going to keep this at 60 degrees C. But at home you could just use a bowl and put some warm water in here from the kettle, mix it with some cold water. You need it to be about 60 degrees C, as I said, so if you've got a thermometer then that's great. So then you would stand your beaker in your bowl of warm water and leave it there for about 15 to 20 minutes. For the next stage of our experiment, you need ice cold, so this has come straight out of the freezer, alcohol. We're next going to take our kiwi solution out of our water bath and give it a little stir, and you need your tall thin glass and tea strainer. So we need about half a glass of kiwi juice, and to that we're going to add our alcohol. We want to make a layer that floats on top of our kiwi juice. So to do that, you need to tilt the glass on its side and then very gently pour your alcohol down the inside of the glass. And then gently place that down on your work surface and watch what happens between the purple layer and the green layer. You will start to see bubbles forming. The bubbles are forming because of the temperature difference between our warm kiwi and our cold alcohol. Your bubbles are stuck in the middle. It's because there's actually a solid substance, a white solid, here, and as time goes on that will become more solid and it will start to float up to the top of the solution, and that is your DNA. There would be some extra stages that happen after this, but what you've done today is take your first steps to become a CSI scientist.